Okay, so picking up, this is now part 4, slide 18. Our bacteria have stuck to our cells. They found the cells they like, or at least they found the cells that they have the right adhesins to stick into the receptors. And now there's all kinds of things a bacteria might do. One thing it might do is inject our cells with proteins. And this is on page 423. This pictured here is what's called a type 3 secretory system, also known as an injectosome. And the term injectosome really describes what it does. The bacteria sticks to our cell, and then it pokes this kind of needle through it, and then it squirts stuff into our cells. These type 3 secretory systems are common in gram negatives. There's many different types found. Type 3 is the one that's mentioned in the book. The kinds of things they squirt into our cells are often proteins, and they are often these things we call effector proteins, or effector proteins. They're called effector because they have an effect on the cell. Typically they induce some kind of change within the cell. They may alter the cytoskeleton structure. So if you recall back to your cell, your eukaryotic cell structure, there's the nucleus, and there's endoplasmic reticulum, and there's mitochondria. But in amongst all that stuff, there are the kind of the girders and the pillars, all the structural elements called the cytoskeleton, made up of microtubules and microfilaments. They give the cell structure. They're also very important in the movement of materials around the cell. Nothing is just floating randomly, generally, in a cell. Things are typically attached to one of these cytoskeleton elements, and then it's transferred from point A to point B inside the cell. By messing up our cytoskeleton, we'll look at an example in a minute. Whoops. They can aid their attachment, and they can cause cells to uptake. They can cause host cells to uptake the bacterial cells. So on to 16.6 on page 423, getting inside the body in the first place. Skin, very difficult to penetrate. It's a pretty inhospitable environment in the first place. It's typically dry, typically salty, and the outermost layer is dead, and those outermost layers are continually being sloughed off, and they are removing bacteria through that mechanism. It's also pretty thick stuff. So some bacteria simply rely on the fact that your skin gets damaged. And a bacteria that's living on your skin, if you cut your skin, that bacteria can then bypass all the defensive barriers and get into your tissues. And that's where it wants to be. It wants to be in your tissues. That's where that's where the money is in our bank. And it can then start to steal your resources and build more copies of itself. Some bacteria have they've gotten together with arthropods and other organisms. Yersinia pestis, that's the one that is transmitted by flea bites. They rely upon the flea to penetrate the skin barrier and inject the bacteria directly into your tissues. Mosquito bites do the same thing. Some viruses like rabies, they cause changes in behavior. They make example, a dog with rabies is going to be more violent, more prone to biting. It's also going to make lots of saliva, lots of virus-filled saliva. So therefore a bite from a rabid dog, first of all, it's more likely to happen, and second of all, it's going to introduce lots of virus into your body. So skin, there are some ways that pathogens can get through the skin, but in general, most pathogens are going to target the mucous membranes. They're thinner, they aren't as inhospitable, they are the entry point for most pathogens. So this goes back to slide 18. We've had a bacteria, it's landed and stuck to the host cell, it's injected some kind of protein. And in this case, in the case of Salmonella, Salmonella uses this type 3 secretion system to inject a protein, and that protein then works to rearrange the molecules that make up your cytoskeleton. It causes this thing called membrane ruffling. And you can see here the effects. Here are ruffles, here's the bacteria itself, and you can see here the mechanism of all that ruffling causing a disruption in the cell membrane and allowing the uptake of the bacteria. In this case, Salmonella wants to invade your cells. This ruffling triggers endocytosis. That is the uptake into a cell inside a membrane-bound vesicle. Okay, so another mechanism that bacteria can have to foil our defenses is here on slide 21. They take advantage of our antigen sampling processes. So in various places in the body, one place in particular, the MALT, the mucosa associated lymphoid tissue, our body, kind of our immune system, is very interested in what's going on outside our body. And once again, if you think about the inside of your intestinal tract, that really is outside of your body in most important senses. So there are places called Peyer's patch in the digestive tract, and their job is to see what's going on, to pull in material from the interior of the intestines across the barrier, and the idea is then that anything that's pulled across the barrier is destroyed. The pieces are then processed by things like dendritic cells and maybe presented to all the various cells of the immune response, T cells and B cells, in lymph nodes, for example. There's a cell in a Peyer's patch called an M cell, and its job is just that. Its job is to uptake material from the lumen of the intestine, the interior of the intestine, and deliver that material to a macrophage. The macrophage is then supposed to destroy the material. There are other ways that the body is sampling. There are dendritic cells reaching their arms through the gaps in the cells and picking up stuff. Our Shigella bacteria, it actively wants to be uptaken by our M cells. It wants to be delivered to a macrophage because it has all the tools necessary to kill that macrophage and escape. And all of the hard work of moving the Shigella from outside the body to inside the body is done by the body itself. Once it escapes the macrophage, it will then penetrate from the back of the epithelial lining of the intestine, and it'll swim around inside the cells. It'll duplicate, and it'll even move from cell to cell to cell. There are many kind of cunning strategies employed by bacteria to get inside our cells. Tuberculosis, for example, produces a protein that causes it to be uptaken. It avoids macrophages in the case of tuberculosis. Let's look back at our Shigella. Our Shigella and Listeria are two very similar bacteria. They direct their uptake, they escape the macrophage, and they infect the cells of the epithelium. And if we look at our Shigella and Listeria bacteria moving around inside the cell, now this is sped up, but you can see that through this process of what's called actin polymerization, these bacteria can propel themselves around inside a cell. They can propel themselves with enough force to leave one cell and penetrate the cell next to it. Actin is a protein that is important in the contraction of muscles. It's involved in the motion of other types of cells, and this mechanism is hijacked by the bacteria here. Okay, so end to this section here. Establishing infection, adhesion. What bacterial structures are involved? So adhesins are the name of the molecule that stick to our receptors. Those adhesins are very often on the ends of pili. And I didn't mention this before, but the pili, different pili can also have different uses. The particular pili that are important in attachment are often called fimbriae. Bacteria use receptors on our cell surfaces. Often it is glycoproteins, just like the majority of viruses do, or it might be a glycolipid. Could be other things too. So any structure, particularly a structure that has a sugar, likes to be, it could be a target for a bacteria to stick. Colonization. Once the bacteria sticks, it has to overcome our antimicrobial chemicals. It has to overcome our IgA. It has to compete with the normal microbiota. In terms of competing with molecules like lactoferrin, this battle for iron, they make their own chemicals to grab the iron, called siderophores. To avoid secretory IgA, they have that pili turnover, shedding of the pili, and shed the antibody that's stuck to it. They have antigenic variation. They can continue to change what they look like. It's kind of like a criminal putting on a disguise, and then they commit a crime and they're seen by the security cameras, and then they go and change their disguise so that when their mugshot is on the news, they aren't recognized. Or we can have a molecule made by the bacteria that just goes up and chops up IgA before it can stick. Penetrating our skin, typically the bacteria have to rely on external forces, i.e. cuts, or they have to partner up with I'm saying partner in the sense that it's not always the relationship between, say, a flea and Yersinia pestis is actually pretty bad for the flea too. Typically arthropods, bacteria live in the mouthparts of the arthropods, and the arthropods are breaking our skin. Mucous membranes. These are a kind of a juicier target for bacteria. They're not as tough. They aren't as inhospitable. There's many ways that bacteria can penetrate our mucous membranes. Salmonella, that does it by causing the membrane ruffling. Shigella does it by being uptaken on purpose, killing the macrophage and getting into the cells. And in both cases, those, well, in the case of Shigella and Listeria, those bacteria can then use this actin system to propel themselves and penetrate out of one cell into the other cell. So Salmonella, that is the one with the type 3 secretion system. That's the one that injects the protein and causes the ruffling. Shigella, that's the one that wants to be taken up, and then it can move from cell to cell using the actin process.